Okay, are we there? We're
I see
recording.
it.
Okay. All right, we are going to get started in a moment. We're going to give it a moment for folks to come on in. Let me just share my screen. And okay, let's see who is here today. Give me one moment. I see Jack and Simon are here and Emily's here. KL is here. This is great. Okay, here we go. All right, so hi everyone. Today is our first day of our spring series of our Teen Science Cafes. So it's really great to kick off a whole new series that's going to bring us all the way through June. For those of you who don't know me, my name is Lauren Traster, and I am the Teen and Leadership Program Coordinator for the 4-H program, which is part of UVM Extension. I'm going to pop into the chat right now. If anybody needs our closed captioning services today, just click on the link that I just put into the chat box. And for those of you that are joining us, we always like to just do a welcome. So find your chat box and tell us who you are and where you are from. So I've already seen that Emily's here from New Hampshire. Simon and Jack are back with us from Montpelier. KL is our friend from Maryland. Welcome. I know a few others have joined us. So just type on into the chat box, do an intro so we know who you are, where you're from. It's always fun to know where everyone's coming from. Okay. Oh, hi, Anna from Essex. So glad you could join us today. All right, so keep introducing yourselves into the chat box. I'm just going to remind you what our protocol, I can't talk today, what our protocols are for when we're here in Zoom. So all of you are muted today, but as you know, that does not mean we don't want you to share your thoughts and ideas. And we do that two distinct ways. So we have the chat box, which you're all using right now. And I see Sage has joined us from Maine. Welcome, Sage. So we use the chat box and we use the Q&A box, and we use those two very differently. So the chat box is used to share your ideas on today's topic, respond back and forth with our presenter. That's how you're going to communicate with her when she asks you a question or wants you to take a guess about something. And then the Q&A box is where you're going to put your questions for her, because we're going to answer all your questions, but we may not be able to get to them right away. So we put them in the Q&A box so that they're there for when we're ready to take questions. And the cool thing about the Q&A box, you have the ability to upvote, which means if you like someone's questions, you can hit the thumbs up, and it just means more people are also like that question, are interested. It moves the question up in the rank, but again, we answer every question that gets posted. So, you know, don't worry where your question lands, because it will get answered today. So as always, I know you guys are a great group. Just want to remind you to be courteous and respectful of one another. I know it's usually never a problem. And just don't create any distractions. Usually the distractions happen in the chat box. So please make sure that what you're putting in the chat box is only related to today's topic. So stay engaged, participate fully. I know you guys always do a great job with that, so not worried. All right. Before we get to today's topic, I do want to remind you all of some upcoming events, programs that we have going on through the 4-H program. So as I mentioned, today kicks off our spring series. The website listed on the bottom of this slide, you can go find this flyer that lists the entire series that's going to be going on this spring. And you can register for each of the different cafes now if you're ready to commit. Hopefully you'll come and sign up for a bunch of the cafes. We also have another Learn to Code program that's going to start next Thursday. This time you're going to be learning how to do some animation. And so you can register for that. It's 6 sessions, Thursdays from 7 to 8 o'clock at night, with a weekly activity that you would do on your own at home to move yourself along for a final project where you're going to create your own custom animation. So pretty cool. I just posted a new program. There is a program through National 4-H Council called Citizenship Washington Focus. This is usually a trip to Washington, D.C., but obviously with the way things are going in the world today, we can't travel. So this is going to be a virtual conference, but it's still going to be really cool because you have the ability to meet kids from all over the country. And the conference focuses on leadership and citizenship. In 4-H, we are offering 8 full scholarships to attend. So this is an application. If you think this is something you would want to do, it's going to be 5 Mondays this summer. And the dates are on the flyer. You can check it all out. But if you think that's something you might want to do, I would encourage you to apply and see if you can get one of those 8 spots. Also, go to this website that's listed because some of my colleagues are also offering some really amazing programs. There's one on herpetology coming up. There's a cool one, a career exploration next week that's going to be highlighting a vet who also does acupuncture in her practice. So that one sounds really cool. There's a gardening program that's going to be starting. And then there's a whole bunch of others. So I would encourage you all, go to the website, see what other programs you could get involved in, because they all sound really good if you ask me. Okay, so let's focus on today's presentation. We're going to be learning about how does Vermont spring snow melt, which is happening right now, even though there's a storm coming tonight, we're going to get more snow. But how does the snowmelt period affect the soil ecosystem? And our presenter today is Brittany Lancelotti. And Brittany is currently finishing up her PhD in natural resources from the University of Vermont Rubenstein School. She is very interested in how fluctuations in soil conditions influence soil nitrogen cycling in riparian areas. So Brittany studied the nitrogen removal dynamics of septic systems at the University of Rhode Island before studying soil biogeochemistry at UVM. That was a mouthful. So I know Brittany's going to tell us all about her work and even some of her, pathway to this work, i.e. biogeochemistry. So let's welcome Brittany. And we are so happy to have you with us today.
Thanks, Lauren. I'm just going to share my screen. And put the chat. Okay. Cool. Well, thanks everyone for coming today. This is a really nice change up to my day of writing to get me more excited about what I'm doing. I can get lost in that sometimes when I'm writing my really long dissertation. So I'm excited to talk to you guys today about a chapter from my dissertation, and that focuses on nitrogen cycling during the spring snowmelt period, which, is happening right now. So I'm actually going to start by asking you all a question just to see what your interest in scientific research is and what are you hoping to learn today, things of that nature. If you guys want to just type in the chat to get the juices flowing, and then I'll go over a little bit about how I got here doing a PhD and all of that. So
I will tell you, Brittany, there's always a slight delay while they're typing, but hopefully you all are typing into the chat. Oh, Jack says advanced virtual reality.
Interesting.
Yeah. Anyone else? Why are you interested in learning more about scientific research? Or what are you hoping to learn today?
Yeah, could be anything, just thoughts.
I know for me, I'm a big skier, so I'm up in the mountains a lot, and I'm always really fascinated by, that whole change in the environment from that sort of winter climate, down all the way down, even now, in the valley, there's no snow, but up high, there's a ton of snow, and the change in that ecosystem from the valley floor up into the mountain, and just how significant that is for, when it doesn't happen, like we had a low snowpack year. So I know some years it's much higher than we had this year. So it's always interesting to see how that change in snowpack affects things too.
Yeah, definitely. Okay, we can move on to, anything and everything about science, but especially language, culture, neurology, super cool. Neurology is really fascinating, I think. All right, I'll just start kind of going over this, and as things roll in, I can address them. Okay, so I went to college at UVM, so I went to undergrad at UVM and I studied environmental sciences. And at UVM, I started working in this laboratory that actually processed wastewater with this natural treatment method, with these like tanks of water and different vegetation in them, which I thought was super cool. This idea of like using the natural environment to do this really necessary process that we need. So I got really interested in research, and I was like, I think I want to do this after college. So I graduated from UVM, and then I applied for a master's to get into a master's program, and that's like a 2-year program. And I'm from Rhode Island, and I just happened to connect with this professor at the University of Rhode Island who was doing other wastewater treatment work. So it worked out super well. And yeah, I loved doing that. And so I wanted to just continue doing research, which I also think I want to do as my career. But now I'm at UVM again, as a crazy coincidence. And I'm studying environmental science, and I'll get more into the specifics of what I study. So yeah, I'm a 4th-year PhD student, which means that I'm graduating soon. Yay. It typically takes like 4 to 6 years to do a PhD. So I'll be done relatively soon. So I study the soil environment, like Lauren mentioned, and processes that transform nutrients like nitrogen. So most of what I talk about today is going to be really focused on nitrogen. Other nutrients include like phosphorus or carbon. Those are really important in the environment as well, but you kind of have to zoom in on one for your dissertation. Otherwise, it would be like a crazy amount of information. So yeah, nitrogen cycling is really important because nitrogen and other nutrients too can be harmful to lake and ocean ecosystems, and that can affect water quality. And so I'm focusing on one part of my dissertation today that involves the spring snowmelt period and riparian soils. So I'll get into what riparian soils are in a bit. So then another thing I want to ask you all is, what comes to mind when you think of the connections, or maybe you've not thought of this before, but looking at these two pictures, what comes to mind when you think of connections between the soil and lakes or oceans? This is a picture of Lake Champlain, where we are in Burlington, and a lot of what I study is like in the context of Lake Champlain specifically, but this applies to any water body in the world.
So KL said sediment, and Simon says photosynthesis. Anna says runoff where the soil can't absorb more water.
Exactly, yeah. Definitely on the right track. So runoff when soil can't absorb more water is definitely along the lines of what I study. So that would be kind of like a flood situation. But also just when the soils aren't flooded, they play a big role in that connection too. So let's see another one. If there aren't a lot of roots, then the soil gets washed away much easier. Yeah, that's very true. So getting back to this connection here. So although they do store nutrients, lakes and oceans, they are sensitive to nutrient concentrations, particularly nitrogen and phosphorus. And we talk a lot about phosphorus when we talk about Lake Champlain. So I'm sure you may have heard about that, or maybe there's a large lake or body of water where you live that suffers from some water quality issues. So soil is really cool because it's like this huge storage container for nutrients like carbon and nitrogen and phosphorus. And so it can kind of help prevent those nutrients from entering water systems. And because of this, they kind of like balance the amount of nutrients that are coming from, or water that's coming off the landscape and going into the lakes and oceans via streams and rivers. So yeah, I think you all are familiar with kind of the framework that I'm working within. So this is a picture of Lake Champlain, probably in midsummer or late summer when things are really warm and you really want to go swimming, but you can't. And so I'll just kind of go over how this works. So basically, nitrogen and phosphorus are what are called limiting nutrients. Has anyone heard of this term, limiting nutrients, before? If you can just type it in the chat. I'll wait for a few seconds. Okay. So limiting nutrients are basically nutrients that in the input or output of those nutrients cause changes to ecosystems. So that's a pretty broad explanation, but specifically, so when we're talking about nitrogen and phosphorus in lake ecosystems, and even ocean systems too, if we give the lake a lot more nitrogen and phosphorus than it's used to having, the cyanobacteria or the algae in the lake can use that as fuel to grow and multiply. So the system is really sensitive to those concentrations. So if we give it a lot of food, they're gonna just go crazy and grow and multiply, like this image we have here with this really green, kind of nasty looking water. So I think there was something else I was going to say about that, but I can't remember. Yeah, you learned about cyanobacteria. And yeah, the unfortunate thing too about cyanobacteria is that they give off these toxins in the water. So it's dangerous for us to be swimming in the water. It's dangerous for dogs and other animals. And basically that can the cyanobacteria being in the lake can disrupt the oxygen concentrations in the water. And that affects fish and things that eat fish and the things that fish eat. So you can just kind of imagine this like cascade of effects that the disruption of oxygen would have on the ecosystem. All right, so where are all of the nutrients coming from? So I kind of have a giveaway here, but does anyone know another source, maybe besides agriculture, that these nutrients like nitrogen and phosphorus could be coming from? Lawn chemicals, yeah. Methane. So methane is a gas, which is definitely, it's a greenhouse gas, so it's definitely a disruption to the ecosystem. But I was thinking more of like solid materials that can make their way into streams and rivers. So, all right, I'll just get into this now. So there are two kind of general forms of pollutants, and that's one, non-point sources. So these are originating from really widely dispersed activities like agriculture, or just forests naturally have nutrients in them that can make their way into streams and rivers. And like stream bank erosion, that can also be a big culprit of like carbon and particulates getting into the stream. So there's not really a way to pinpoint and say, oh, it's coming from this area within this watershed. So that's a non-point source. And point sources can be wastewater treatment facilities. So like the big municipal wastewater treatment facilities that you might have in your town or city. So those serve all of the homes in the area. Versus like if you have a septic tank at your house, that would be more of like a non-point source, because there are so many of those that we can't really trace which one it came from. And then other things like stormwater. If you're thinking of like a big pipe, a stormwater pipe that's leading into the lake, that could be a point source. So a lot of the time when we're talking about nitrogen and phosphorus pollution, we usually talk about it in the context of agriculture here in Vermont, because we do kind of know that it is a large culprit compared to other sources. And it's kind of interesting because we have the Vermont, I keep saying we, but we're not all in Vermont. Vermont has the largest agricultural economy in New England, but it's a relatively small part of Vermont. So only 14% of Vermont's land is used for agriculture, but the way that, or where the farms are located, are really making them contribute a lot to nitrogen and phosphorus pollution. So I'll show you this map here. And so this is a map that the EPA put out, the Environmental Protection Agency. And so this is Lake Champlain here. And all of these areas in yellow are agricultural lands. So, I mean, it could be like active or non-active farms, but the point here really is that if we combined all that yellow space, maybe it wouldn't be a very big space, but it's located so close to the lake that unfortunately has a big effect on water quality. So I wanted to ask another question, which is, before I kind of get into how we address this problem, does anyone want to take a guess, or does anyone know anything about what we do to kind of mitigate these problems in Lake Champlain or even other ecosystems, maybe where you live? I'll pause for a second here to give you time to think.
So Anna says plant borders around farms to absorb nutrients.
Yeah, so that's in a way very related to what I'm going to talk about, stream buffers. Yeah, buffers and borders kind of gets into the concept that I'll be talking about.
Kale, you say other individual actions. Is there a specific one you think an individual could take? Or if anyone else thinks of an action an individual could take? Limiting the amount of fertilizer. Yeah.
Definitely. That's an individual action, or it could be like on a community scale too. Okay, awesome. So what's being done to solve the problem? So in the US, we have the Clean Water Act. Oh, we have another, farmer. I'll talk about that too, what farmers can do to mitigate this problem as well. So in the US, we have the Clean Water Act, and you've probably heard of this before. So that requires states to basically ensure that the water quality of their water bodies are met for each, lake, stream, river in their state. And so that's really to ensure this healthy habitat for fish and shellfish and wildlife, and so that we can recreate in these areas. Yes. Okay. So we have what are called these nutrient reduction strategies. And one main one is a total maximum daily load. So those are established because we want to understand how much pollution or nutrient pollution we can actually put into the lake without causing algal blooms and all of those ecosystem disruptions. And so apart from that, we also have in Vermont specifically, but of course other areas around the world as well, we have the 25-foot riparian buffer requirement. So I'll get into more of what riparian buffers do, but specifically in Vermont, the Agency of Natural Resources has implemented this standard, that 25-foot buffer. So I'm going to pause again and ask you all if you've heard of riparian buffers before I tell you more about them. So maybe if you've heard of them, or what do you know about them? Before I kind of get into this, there's this picture here for you, but just
Kale
curious
says
to see.
definitely.
Definitely.
Kale, I knew you would know about the riparian buffers.
It seems like it's something that's pretty well known, especially in Vermont, because we have that like agricultural source of pollution, and there's kind of been a lot of studies around specifically the ag.
And I bet more than one person has probably done a service project where they maybe have done some planting along stream banks and stuff like that to help with it.
Okay, let me just get into this a little bit. Oops, I didn't want to go to this slide yet. Okay, so riparian buffers, although I think a lot of you know what they are, but I like this image here because it shows the cornfield where fertilizer is applied, that source of nitrogen and phosphorus. And it shows this sort of transition zone over here before we get to the stream. So that transition zone is super important to like slow down the water. That's if it's flowing over the surface, maybe in like a big rain event, it can slow down that water. And when it slows down, it can seep into the soil, and that's when things start to happen underneath the soil that can transform the nutrients and kind of make them just stay in place there, versus just if you could imagine, if you dumped fertilizer on here and this part wasn't here, you'd just kind of be dumping right into the stream. And so more specifically, so now just zooming in on nitrogen, there's a few things that riparian buffers can do. to reduce nitrogen inputs to streams. So the first thing is sediment trapping, kind of like what I just talked about. So yeah, just trapping that water that probably contains lots of sediment inside that riparian buffer, because there are so many different like trees and roots and plants that are kind of an obstacle that block the water from getting all the way into the stream. And also plant uptake. So riparian areas are habitats for lots of different plant species and trees, and plants need nitrogen and phosphorus to grow and thrive. So they are taking those nutrients up from their roots as well. And then we also have this element of the microbial uptake. So microbes also need nutrients to grow and multiply, and they're going to be a source of uptake as well. And then we have this process called denitrification. So I'm curious if people have heard about this, because this is really what my dissertation is really focused on. And I kind of, when I ask people or when I tell them about my research, some people kind of know what it is, but I feel like most people have not necessarily heard of it before. So if you want to type in the chat, maybe yes, you've heard of it, or no, or maybe a guess of what it is.
So Kale says not under that name, but nitrogen in the water is a problem here in the Chesapeake Bay area where he is at.
Yeah, definitely.
Anyone else have a guess of what denitrification is? Denitrification. What do you think? I have a guess, but I'm not going to say it because I want one of you to guess.
Nitrogen extraction.
Nitrogen extraction from soils and stuff.
Definitely on the right track. All right, we can move on to the next slide. So yeah, so denitrification is a way that nitrogen is removed from the soils. And the key part, which maybe if you kind of use the context clues to figure out what it means, you maybe didn't know that it's this microbially mediated process. And so this little illustration here is kind of cool. Although we wouldn't be able to see microbes like this, it kind of, yeah, shows the diversity of the different microbes. And some of these guys might be what we call denitrifiers. So microbes that can denitrify. So I had, this other question. What comes to mind when you think of soil microbes? Like, have you learned about them or researched them on your own, or like, do you ever think about soil microbes? Because we can't see them, and they also play a huge role in life on Earth, which is kind of cool, even though we wouldn't know they're there. They make soil. That is soil, not dirt.
That was me trying to say they make soil, soil, not dirt. It's hard to type. It's hard to type that. The emphasis.
Yeah. Yeah, that's a cool way of thinking about it. Like, they make soil because they do all these processes that soil is that's unique to soil, basically. All right. So one key thing here that is pretty crucial to kind of understand as we move further in the presentation is that soil microbes can get their energy from transforming nutrients from one form of a nutrient to another. So it's basically a win-win situation because they thrive and survive off of transforming nutrients. But we on Earth, we need them to do this process because we're kind of pumping these forms of nutrients into our soil that wouldn't necessarily be there if we didn't fertilize our food, and all of these processes that we do on Earth that were not naturally here before we got here. So I'm going to show you a little schematic here that kind of represents this transfer of one type of nitrogen to another. So first we have nitrate, and this is the molecule that I mentioned before that is like fuel for cyanobacteria. But this, you know, we don't want this in our water bodies. We don't want this in like large concentrations in our lakes and streams and oceans because it disrupts the whole ecosystem when there's an algal bloom and the oxygen concentrations get all messed up. But this N2 molecule, nitrogen gas or dinitrogen, is actually like almost 80% of our atmosphere. So it's completely inert, or that means kind of like harmless. So microbes can actually turn this into this, which is perfect for us. That's basically what we need. And they do this by reducing this molecule. So they're kind of stripping off these oxygens here and all the way to N2. And so you might notice here that we have N2O, nitrous oxide, which is not great because it is a greenhouse gas. So sometimes this process does make nitrous oxide if it doesn't go to completion. And that's due to various conditions in the soil. But basically, the gist is that we're converting this form that can cause algal blooms to this inert or harmless gas. And that's actually, fun fact, that is actually how we treat our wastewater. Like a septic tank is going to have, it's going to be this ideal environment for this process to occur because there's lots of nitrogen in wastewater. So getting kind of deeper into denitrification, there are some requirements that we need for denitrification to happen naturally in the soil. And so those are organic carbon, and then of course we need a nitrate source. And there are some regulators of this process. So soil pH, ideally it would be like neutral, 7 to 7.5. Oxygen availability, we need really low oxygen concentrations for denitrification to occur. And we also need like a moderate soil temp. This is like 40 degrees Fahrenheit, 5 degrees Celsius. So this is, as a scientist, one of my big questions is like, how do all of these different elements interact to affect denitrification rates? Because we want to know where is denitrification going to happen and when that's going to happen, because we rely on this process to help us mitigate our nutrient loading problem, especially in Lake Champlain. So now kind of connecting the denitrification and the riparian buffer stuff. So the reason why I'm studying riparian buffers is because they have the capacity to be great habitats for denitrification. And so basically what's happening here is this illustration is kind of showing you all of these different hydrologic flows, which is just like different water flows from rain that's coming down or groundwater. So underneath the soil surface, there's groundwater stores that kind of fluctuate. That's kind of like what these arrows are representing here. So there are all these different flow paths that are kind of converging together. And that's because of where the riparian buffer is situated in the landscape. So they're usually in these low-lying areas that receive a lot of water. And so that makes a really good habitat for denitrifiers, because that can increase substrate availability. It can drive up soil moisture. And usually when we have high soil moisture in the soil, we have low oxygen concentrations. And so if we think back to our elements that are modulating this process, if we have high substrate availability, we probably will have a good amount of nitrate, or this is a molecule called nitrite. They're both can be used for denitrification. And we also have our organic carbon. And then if we have that high soil moisture, low oxygen, we have good oxygen availability to promote denitrification. Okay, so now that I kind of told you that, thinking about the spring snowmelt period, like right now in the mountains, it's probably still occurring. This heavy snowpack is melting. How do you think that would be a good time for denitrification to happen? Or maybe you don't think that, and why? If you want to enter into the chat and increase the soil moisture, yep, definitely. Yeah, wetter soil for sure.
And Anna says increases soil moisture, which same with Al saying
Yep.
wetter soil. Okay, Al says slower movement.
Slower movement of like nutrients, do you mean? Okay, I'll just get into this now and see if more responses roll in. So yeah, spring snowmelt, especially for us in Vermont here. So we have, where I am, we're in a valley, and we have the green mountains all around us that build up these huge snowpacks. And so in an area like this, snowmelt is going to be a really important hydrologic event. And that's because it's melting and going to go down slope into our streams and rivers and eventually reach Lake Champlain. So this can account for the majority of annual stream flow. So annual stream flow is just going to be like the sum of all of the water that gets into the lake. So it's basically just accounting for a ton of what ends up in the lake. And so the mobilization of nitrogen during spring snowmelt has been linked to those algal blooms in some systems. So we're not quite sure if this is directly related in our watershed here in Vermont, but in some situations, it's been shown to really affect the summer algal blooms. And so this is a really important time for removal of nitrogen and retention of that nitrogen. So this is kind of going to illustrate what some of you said about soil moisture. So how do the soils change as the snow is melting? Okay, so that's my illustration of how the snow is melting. And that's really going to wet up the soils. They might even get so wet that they can't take on more moisture. So in a situation like this, oxygen levels are likely going to decrease and substrate availability is going to increase. So again, we have our nitrate or our nitrite, that's a check for denitrification. Organic carbon we have, that's a check. And that oxygen availability is probably going to be low. So those are all great for denitrification. And that's a good thing, right? Because we are concerned with lots of nitrogen being mobilized into our rivers and streams. But if this is a good time for nitrogen retention and removal, then that's great. So my research kind of investigates that, is the spring snowmelt period actually a good time for denitrification? Okay, so now I'm going to get sort of more into the study that I'm doing. So I'm going to introduce you to our riparian area study sites that we have here in Vermont. And so our first one is our agricultural site, and that's in Hungerford Brook. So that transects Hungerford Brook. You can see that kind of back here. So we sort of contrast a lot of the time when you do research like this, you want to sort of replicate it in two different scenarios so that you're repeating your experiment, but you're looking at it in two different perspectives. Like, how could these variables affect this mechanism here as opposed to in another place where the conditions are different? So here at the ag site, we have high nutrient inputs from fertilizer. The farmer fertilizes this field and feeds his cows. And then this site is lower in elevation and we get less precipitation than we do at the other site I'm going to show you. And a lot of the vegetation here is grasses and nettles. So I must have taken this in the spring or winter, but it's usually you can't even really walk through here without being covered with all sorts of stuff that you don't want to be covered with. Then we have our forested site, which is transecting Wade Brook. And at this site, we have lower nutrient inputs because there's nothing really going on around here. People live around here, but it's very, very scarcely populated. It's higher in elevation, so we get more precipitation and more snow. And there are lots of trees, like maple and beech trees. And then I just put my dog here because she loves this site. And that's a really fun part about my job, is that I get to bring her sometimes. And so both of these sites are located within the Missisquoi watershed, which I'll show you on the next slide. So here's the Missisquoi watershed up here. So it takes us about an hour and a half to get up there from Burlington. And yeah, you can see how that would drain right into Lake Champlain. Okay, so our study sites are set up kind of like these transects here. So this is the stream and this is a transect. And at each of these little dots here, we have a bunch of instrumentation. So we have a bunch of soil sensors. And we set these up in this way because if you can visualize here, sort of this gradient here of the riparian area, the water is going to be moving down here like this way into the stream. So we wanted to kind of capture along the way what's happening. So what would the water encounter as it's kind of moving towards the stream? And that's repeated at the forested site. So we have a bunch of sensors. And so what I do here is I monitor the soil conditions with the sensors, and I also measure how many denitrifying microbes there are. So the microbes that do that denitrification process. And I also want to know how much denitrification is occurring. So here's a little schematic of my experimental design for this project. So here's our forested site and our agricultural site. And at each of these sites, we have an upland area and a wetland. So upland is going to be pretty dry. It's well drained. So like if it rained, the water would just seep into the soil easily. Whereas the wetland is closer to the water table. So it's going to be kind of waterlogged most of the year when that water table is high. And we have a bunch of soil sensors, which I don't really need to go into right now, but I'll show you data from them in a bit. And I also collect denitrification rate, and I measure the nitrogen and the carbon in the soil, and also in the snow, in the soil water, and in the stream. And again, I measure the abundance of the denitrifying microbes. So there's kind of like a lot going on that you have to measure to kind of cover all of your bases. But then when you compile all of that data, not all of it is going to be completely relevant and important. So if it seems like a lot, it's usually condensed down to like some core pieces, which I'll show you in a bit. So how do we measure the denitrifying microbes? Well, I guess I was going to ask you if you had an idea, but now that I have this up already, I'll just kind of go into the DNA thing. But if you have any thoughts on that, feel free to put them in the chat. So basically, just kind of like summing it up into a few sentences, we use DNA to measure how many copies of a certain gene is present in the soil. So we can think of a gene as this area along the DNA that is going to encode for the things that are necessary for the cell to, or the microbe to denitrify. So we've kind of figured out that we can measure the abundance of like that specific area of the genome, and that equates to roughly how many microbes there are that can do that process. So I'm specifically, you can do this for anything, but I am doing this specifically to target those denitrifiers. So this has been a really fun part of my dissertation, although it's actually, it can be not so fun sometimes too when experiments don't go super well. But overall, it's been a really interesting and fun thing to incorporate. All right, let me just check how we're doing on time. Okay, so I'm gonna show you my research questions here. So research questions are really important because they guide basically what you're gonna do, what you're gonna base your experiment off of. And a lot of the times they're based on like what already has been figured out and what really needs more research attention and what the science community needs to know more of in order to better understand the system. So that's kind of how you have to do a lot of reading and maybe some prior experiments to really get at what you should ask and what you should target in your experiments. So the first one is, are soil conditions supportive for denitrification during the spring snowmelt period? So technically they should be, but are they? We don't totally know. Do land use, agricultural versus forested, so our two sites, and landscape position, so that upland versus wetland position that I showed, influence denitrification rates? And are denitrifying microbes impacted by land use and landscape position? Okay, so this is the first figure that I'm showing you here. So I'm gonna walk you through what you see, and then maybe you can take a little bit of time and kind of try to interpret what's going on, and then I'll kind of tell you what I notice about it. Yeah, box and whisker plot, exactly. So on the y-axis here, this is denitrification rate. And so this is a box and whisker, like KL said. And I just want you to focus on this middle bar here, because that's going to be the median or the average. And we don't have to worry about the other elements here. I think for this one, we should really be concentrating here on the middle bars. And then on the x-axis is going to be, so this one is the agricultural upland. So right here, this is the agricultural wetland right here. And then the forested wetland, you can see that up here in the forested upland. So we're looking at how denitrification rate is changing according to the different land uses and landscape positions. So if you want to like take a sec and think about maybe if this was like an expected result or not, something like that, then I can share with you what my conclusions were. And don't be afraid to take a stab at it. You guys are really smart, so I'm confident you know what's going on here. Okay, I can start to give some hints here. So I think, let me see. So I put this box around these two because I can really tell that these averages here are larger than these. And so really right off the bat, zoomed out, I can kind of tell that at the agricultural site, the rates were higher than the forested site. And this is sort of what we would expect, just because nitrate is probably going to be higher here. And higher nitrate usually means higher denitrification. Okay, so I'm going to show you a few more figures here. So on the x-axis, we have the date. And up here, this panel is the denitrification rate, just like what we looked at in the other slide, but this is just looking at it in a different way. And then we have soil moisture data and oxygen data. And these bars here, so this is when I went out in the field and collected data. Whereas, so So that's why there are separate bars here for the denitrification rate, whereas the box and whisker are all of those values combined. But yeah, and feel free to like say what you think about these as well in the chat. So this is the forested upland site. So this is a site, if we're just thinking about it really zoomed out, the forest is not going to have a ton of nitrogen because, well, actually forests are usually nitrogen limited, and we aren't really, there's no source of like fertilizer or wastewater that we know of. So this is a really just natural environment. And it turns out that there's not much going on at this particular spot. So that's going to be here, this forested upland. And again, we could see with the soil moisture, this is pretty low. You don't have anything to compare it to right now, but the soil moisture is quite low, and that's driving oxygen up to be really high. So we need oxygen to be like here for denitrification, and it's way up there. So that's not really surprising. So we're looking at the same plots here, but for the forested wetland. So that like low lying position where it's usually inundated all the time. And we could see there's a little bit of activity here. Not much, but right around here in like April, mid-April is when we had some denitrification happening. And you could see at this location that the soil moisture compared to the other location is higher for sure, and the oxygen is much lower. So that's going to be a pretty good area for the denitrifying microbes, technically. And there's all sorts of reasons that we could think of that there's not a ton of denitrification going on here. Like there might be a limited nitrogen supply, and that might be really preventing lots of denitrification to happen. But at the same time, if there's not a bunch of nitrogen in the forest that needs to be sequestered, then that's good. That means that it won't make it to the stream. So yeah, this is kind of what I already went over. So let's just look at the agricultural upland as well. So we can see a lot more denitrification going on in here versus the other site. And we don't necessarily see low oxygen here. This is pretty high oxygen, but we're still seeing some denitrification going on. And so that could tell us that there's some other sort of mechanism going on there, that the denitrifiers aren't really limited by the high oxygen because they don't really care. It's still, somehow it's still going on. And then when we look at the wetland site, still a pretty good amount of denitrification going on here. And when you look at this, you would say, oh, there's probably a ton happening here, but actually we didn't sample at that time. For some unfortunate reason, we missed that drawdown here. And then because of COVID, I wasn't able to do another sampling year, which was kind of a bummer, but that stuff just happens in science and you kind of have to just roll with it. So yeah, we can spend a little bit. I can pause here for a little bit if you want to just look at these more carefully, and if you want to add any comments about these. There is kind of a lot going on here. Like it takes a long time to actually like pull out this information that you that you think you need to understand what's going on in the system. Okay. So we have 10 minutes left, I think.
Yeah.
Yeah, so let's move on because I want you guys to have time to ask questions. So these are the denitrifiers. And here, so this is, you don't really need to know what NOS-C is, but that's just a specific gene that we target. And that will tell us how many denitrifiers are at our site. And basically, there isn't a significant difference between the agricultural and forested site in terms of the denitrifiers. Sorry, my dog is eating her food very loudly. So yeah, that's an interesting point because we're seeing these really like different ecosystems, but we don't really see a difference in the number of denitrifiers that are there. So that's something we're going to explore further. And then if we look at this by land use and landscape position, so we see this high abundance of denitrifiers here at the agricultural upland. So remember, this is where the conditions weren't really great for denitrification, but we still saw a lot of denitrification happening. And this kind of makes sense because we have a lot of denitrifiers there. So that is somehow working for the system in terms of creating these high denitrification rates. There's a lot of denitrifiers. And then in the opposite way, we can see that the forested upland was below detection limit in terms of the denitrifiers present. So we couldn't detect any there. And that really coincides well with what we found in terms of denitrification rates because really nothing was happening. So this is kind of an ongoing study too, in terms of like analyzing the data. So I'm sure we'll have some interesting insight soon once we finish kind of analyzing that and like reading into different experiments that kind of did the same thing and understanding like what our results mean. So not quite fully done with that yet, but I wanted to share this because it's one of my favorite parts of my thesis. So just some concluding points here. We saw the low denitrification rates at the forested site, which is probably because it's a nitrogen limited site, and actually the trees and plants might outcompete soil bacteria for nitrogen. There were those higher rates at the ag site, which is probably because of nitrogen fertilizers that are applied to the field. And we didn't see differences in denitrifying bacteria between the two sites. And it could be because there's not a strong connection between the abundance of denitrifiers and the denitrification rates. So that's something I'll be exploring a little bit more going forward.
Well, thank you, Brittany. That was fantastic. So I want you all to be thinking, if you have questions for Brittany, put them in the Q&A, but I'm going to launch a poll right now. This is our standard poll, 2 questions, just to give us some feedback, how you thought the cafe was today, and then did you learn anything? So 1 to 5, 5 being the highest, 1 being the lowest. And once we get done with the poll, it'd be great if some of you have some questions for Brittany. So just give it a second. Okay, I think we got everybody. Thank you on that. And any questions? At this point, you can Q&A box or type into the chat, whether it's about Brittany's schooling, whether it's about her research, her methodology. I'll start it off. Where are you from in Rhode Island?
So I went to high school in East Greenwich. But now my mom lives in Narragansett, and so I kind of, when I go home, that's where I am. So that's where like the beach is. And
Nice. I went to school in Providence and my husband
oh,
grew up in Ashaway. So right by
nice.
Misquamicut Beach.
Cool. Yeah, I'm actually going to be moving back in May to finish my PhD remotely from there.
Oh, nice.
Yeah, so I'll have the summer there.
All right, so I'm not seeing any questions coming in. I'm going to give you a countdown of 5 because here is a great opportunity. But obviously, Brittany put her email, and I know if something comes to your mind later on, feel free to email her. So why don't we all thank Brittany for sharing her research with us today? Hopefully that illuminated what it means to do some research on natural resources and give you an idea of some of the research that goes on at the university, especially at a PhD level. So Brittany, thank you so much for being with us today. We so appreciate it.
Yeah, thanks everyone for coming. This was fun.